And now
Is
you're,
this okay if I stand over here and then you can see the screen? And
but now you're off camera.
that doesn't matter. You need to see me. You need to see the slide, so that's fine.
And I can move just the camera
So at that
closer.
point, maybe zoom in, Curtis. And stand off.
I can move the thing.
Where's the online stuff? I don't know.
I might could move this closer.
Just a thought.
I think it's better elevated.
Apologies to people online that are watching this.
What about the people online?
Apologies to them as I'm moving this camera around and they're getting vertigo and Teresa's on.
Yesterday we had between 10 and 15
Well, it's not bad.
most of the time.
If I get that today, I'll consider that a moral victory.
You'll have to tell me how I'm doing with the camera here.
Oh, she says there's no sound. Oh, it's 6 a.m. now.
Like,
Who's up?
Definitely not my son or my wife, I can tell you that.
Is that better?
It's not, Rena.
What's that? You said that and then I unmuted him.
But then she said it's not.
Can I get a picture? History of leaking. It was funny, I saw Jeremy and Faith this morning at breakfast. Jeremy asked me, do I have my talk ready yet? I realized, actually, Jeremy, I've been working on this talk for 30 years now, so I hope I have, I've got it ready.
Please mute on her side, turn up her speakers.
I'm prepared for your talk today.
Awesome, you are ready.
Awareness.
Beautiful.
The weird angle. Slides.
Far away to reach the slides and it's got some. And if he's not gonna be on camera anyway.
Now, you don't need my face, you don't need to see my face.
We're about to get started, so if everyone wants to make their way in.
So start with this.
All right, welcome back, everyone. Just a couple announcements this morning. We have a few extra t-shirts. If anyone is interested in purchasing them, we can do PayPal or PayPal through our website, or you can do Zelle or cash. They're $15 and out there. It's just the leftover sizes, so if anyone is interested, you can get those.
Or a check.
Or a check, yes.
You have any hoodies? Or
The
just-
hoodies were only online, they get sent directly to you. We just have the t-shirts
We
here.
have kid sizes.
There were a few kid sizes left, yeah. And tonight is the basketball game, so we're super excited about that. I have everyone's tickets, so thinking maybe a lunch break or sometime today before then, and I will get your tickets if you order tickets. But let's go ahead and get started. So, I'm gonna give you Paul's bio, but we all know a lot about him already, and are very grateful to have him as a part of our organization. But Dr. Paul Grossfeld is a board-certified pediatric cardiologist and clinical professor of pediatrics at the University of California, San Diego. Dr. Grossfeld takes care of patients with congenital heart disease at Rady Children's Hospital in San Diego. He saw his first patient with Jacobsen Syndrome in 1995, and since that time has assembled a multidisciplinary group of investigators studying the genetic basis of congenital heart defects in Jacobsen Syndrome. Dr. Grossfeld serves as the Chief Medical Advisor for the U.S., Spain, and European Jacobsen Syndrome Support Group. He has reached out to colleagues both locally in San Diego and from around the world to bring together a team of clinical experts to help with the various aspects of care for individuals with Jacobsen Syndrome. He has co-authored over 20 peer-reviewed scientific papers on Jacobsen syndrome. His most recent publication describes the identification of two novel cardiac growth factors that are deficient in genetically engineered mice lacking the ETS1 gene, which is the Jacobsen syndrome heart gene, suggesting the exciting possibility of a new therapy for patients with congenital heart disease and potentially in adults suffering from an acute myocardial infarction. He lives in San Diego with his wife Susan and their 19-year-old son Stefan. In his spare time, Dr. Grossfeld loves to travel internationally, play a variety of sports, and swim and snorkel in a nearby La Jolla Cove in the summer. So, welcome, Paul.
All right, thank you. Like I said, it really is so neat to give a talk to you guys because you are the talk. So, like anything else I've ever done it in terms of talks, so, feel free to stop me at any time. What I'm gonna do is actually start out, oh, by the way, are we not doing the subtitles? I forgot about that. Okay, we'll take a pause. Any questions? Feel free.
What is Jacobsen syndrome?
What I will do actually, before I talk about my research, is give a little bit of an overview of the clinical aspects of cardiac defects in Jacobsen syndrome and some clinical recommendations, and then I'll take it back. Our research. I'm glad you know what to do, Curtis, because I don't. Okay, a little harder for me to see from over here. All right, so in terms of the congenital heart defects in Jacobsen syndrome, about half of your children are born with some kind of a congenital heart defect. So even with two deletions, the identical size, one might have a very complex heart defect and one might have a completely normal heart. One of the things that we learned early on is that there is probably an unprecedented high frequency of hypoplastic left heart syndrome. In Jacobsen syndrome, maybe as many as 10% of all babies, and we have two babies here today with hypoplastic left heart syndrome. And that accounts for probably about 1 to 2% of all cases, all comers. In other words, there are about 1,000 babies born each year in the United States with hypoplastic left heart syndrome, and so maybe 10 of those are born with also Jacobsen syndrome. The other thing that's interesting is for the rest of you whose children have a heart defect, it's not hypoplastic left heart syndrome, but it is some other type of heart defect like the VSD that we talked about this morning and many others. So the bottom line is the majority of the most common heart defects that occur in the 1% of the general population with any kind of a heart defect also occur in Jacobsen syndrome. So early on, that was pretty appealing to me to think, well, maybe by studying this rare syndrome, what we learned from that rare syndrome could apply to a much broader population. And that's truly turning out to be the case. We do have some clues as to why your child with one deletion might not have a heart defect, whereas another one with the same deletion might have hypoplastic left heart, or another one with the same deletion might have total anomalous pulmonary veins. I'll maybe touch on that briefly, but that's a little bit out of the focus for today, but we are actually gaining some interesting insights into that as well. Okay, so in terms of some clinical cardiac recommendations, some of this is kind of after the fact for you guys, but for anyone who is diagnosed prenatally with Jacobsen syndrome through the non-invasive testing that you heard a little bit about yesterday or other types of genetic testing prenatally, there should be an automatic referral to a pediatric cardiologist who specializes in doing what we call fetal echocardiogram. So that's an echocardiogram of the fetus's heart, very, very detailed, and that's called a level 3. So if you were diagnosed these days prenatally with hypoplastic left heart syndrome, we don't want you delivering somewhere out in the desert, but obviously at a level at a tertiary care pediatric cardiac center, we can manage immediately the emergent needs that a newborn with complex congenital heart disease needs. Again, if it doesn't happen for whatever reason until after birth or the diagnosis of Jacobsen syndrome is made, we recommend, if it hasn't already been done, pretty much an immediate comprehensive cardiac evaluation, which should include at the very least imaging, usually an echocardiogram. And obviously, depending upon what's found, then the plan is put in place for long-term follow-up. Something that is, we kind of come and got a little bit in terms of specifics, but if your child initially has a baseline evaluation, which is completely normal, it's a little bit arbitrary, but there are rare complications that can develop over time. So if they're doing well and they've had a normal echocardiogram, my recommendation at this point is maybe just kind of a follow-up surveillance assessment, which should include a repeat echocardiogram about every 5 years. But obviously, if there are any new symptoms that could be suggestive of a heart problem, that would require immediate referral, things like fainting, palpitations, signs of heart failure. I should have put on there a new heart murmur, even though most heart murmurs are normal. You want to make sure that something hasn't developed there. Unlikely, but still potentially possible. Okay, so I actually kind of gave this talk a few months ago at the European conference in Germany, and they were kind of my guinea pigs. So you're getting the revised and improved version of it today, hopefully. And I thought rather than throwing out all of this hardcore science at you, I would kind of start by giving you the punchline of what our research has accomplished. I'm going to try to come over to this side. Yes. All right, let's see. Forgive the fine print here. I know it's a lot of information, but as I mentioned, about half of your children with Jacobsen syndrome have some kind of a heart defect. You've heard already in the last few days, the ETS1 gene, which is the first gene that we think is a major contributor to the heart defects. And actually, it turns out right next to ETS1 on chromosome 11 is sort of a sibling of ETS1 called FLY1, which you've also heard about. And I'm not going to really have a lot of time to go into it, other than to say that we now have compelling evidence that both ETS1 and FLY1 together, when lost, are what causes hypoplastic of heart and some of the other heart defects in different combinations. So that's kind of a relatively recent finding. And I've just spent $23,000 to have a company genetically engineer a mouse to have those two genes together deleted so we can study that. Okay, just some other things to summarize that I'll come back to in more detail is when you want to learn about what the function is of the gene, you need to know where and when. So my lab was genetically engineered mice whose hearts are actually very similar to a human heart, and the genetics are very similar. So basically, most of the genes that we talk about for a human also occur in mice, and we have some amazing technologies that I'll touch on to help us to study exactly what these genes are doing. So one of the first things we wanted to do is to ask where and when is ETS1 expressed, and I'll show you that in a few minutes. Basically, as the heart develops, there are a lot of different populations of cells that have to work in a very coordinated, amazing way, both temporally and spatially, to form a heart. And two of those populations of cells or lineages are what's called the neural crest, and I'll talk about that in a minute, and the endothelium. So those two populations are where ETS1 is functioning. And how when it's knocked out, it's affecting how those cell lineages work to form the heart. Interestingly, though, like with hypoplastic left heart syndrome, the big problem is the development of the actual heart muscle. So what we call the myocardium. And neither of these genes are actually expressed in the myocardium. So some other population of cells that's actually regulating the myocardium, or what we call the cardiac myocytes. And I'll tell you more about that in a minute. And actually within the heart, what we call the cardiac endothelium, there are actually two populations. There's one called the endocardium. That's a population of cells that lines the inner part of the ventricle. And then there's the other endothelial cells that actually make up the blood vessels that supply blood to the actual heart. Those are called the coronary arteries. So we call that the coronary vascular endothelium. And I'll come back to both of those points a little later on. And very interestingly now, both in our mouse models and very recently in a human Jacobsen syndrome hypoplastic left heart, we have found that these populations are decreased. And I'll come back to what that actually means. So in a human heart with hypoplastic left heart syndrome and from a baby with Jacobsen syndrome, now we see a dramatic decrease in those coronary vascular cells that are required normally to supply nutrients to the growing, developing heart. And I'm probably not going to touch too much on this, but I collaborate with a few amazing principal investigators, PhD researchers that use other animal model systems, and we have used these different animal model systems to study these genes. And what we're doing is looking for common sort of mechanisms across species that enlighten us as to how these genes work. Okay, so Curtis, do you think we can turn down a little bit the lights like you did yesterday or Monday? That would be really nice. Come on in, John. See, the problem is I know most of your names, so I can call you out or someone's nodding off. All right, so thank you, that's perfect. So this is kind of your garden variety cartoon of a hypoplastic left heart. So, by the way, can you guys see it or am I blocking it? Okay, well, let's see if I can set that, sorry. So on the left is just a cartoon of a normal heart, so you can appreciate the chambers, you can appreciate the valves, you can appreciate the aorta, the red vessel coming out to supply blood to the body from the left ventricle, and the pulmonary artery supplying blood from the right ventricle to the lungs. And what you can see in this diagram is this is typically what you see when you go to the internet. A hypoplastic left heart is characterized by, oops, that's not what I did, is characterized by a very diminished left ventricular chamber. And in this case, what it appears to be is there's like this thickening of the wall of the ventricle, the myocardium, so it sort of grows inward and obliterates that actual chamber volume. And intuitively, what it means is that that small volume left ventricle now is not able to pump enough blood out to the body to meet its needs. And that's why without emergent imminent intervention right after a baby is born with hypoplastic left heart syndrome, they will not survive for more than typically a few days. So that's kind of your schematic diagram of hypoplastic left heart, but like so much in medicine, it's much more complex than that. What in the real world, I should say, I'll connect to that. In all comers with congenital heart disease, hypoplastic left heart syndrome still continues to be the #1 cause of death. So it's quite a challenge. Economically, I don't think I have to explain to you guys the burden it's put on you, even in the best of circumstances. Probably well in excess of $1 billion a year, not to mention all the indirect costs, emotional stress, time lost from work. So it's a big, big problem. And if you come to Rady's Hospital, even though like I didn't tell you this, maybe about 3 to 5% of all babies with congenital heart disease have hypoplastic left heart syndrome, but because of the morbidity and all the complications, almost at any time among our 30 or 40 patients in the hospital, there's always going to be a few with hypoplastic left heart just because of the complexity and long-term challenge. Okay, so I was alluding to this before, and that is, I showed you the schematic, but in the real world, there are actually different versions of hypoplastic left heart syndrome. There's actually classic, and that's what these 4 pictures show. And there's even more versions that we call atypical or non-classic. But what I want you to do is just focus on these 2 pictures on the left. So this is one form of hypoplastic left heart syndrome that does kind of resemble that cartoon that I showed you, where this is all that thickened wall of the left ventricle, the thickened myocardium, and this little thing here is what was supposed to be the chamber volume itself, the cavity. And this white is actually scar tissue that typically goes along with this. But in this case, even though we call it hypoplastic, I don't think it's a hard sell to convince you that this is actually overgrowth, right? It's too much growth. And you would be shocked if I told you how, unfortunately, what set this field back so much is for the few PhDs, let's say, that have been studying this heart defect, they're going to say that hypoplastic left heart syndrome is due to decreased growth because it's called hypoplastic. Decreased growth, really? So my colleague in children's, the head of our pathology department, came up with a good term. She thinks we should actually call this hyperplastic left heart syndrome, right? Because that's really the mechanism. What we found in Jacobsen syndrome is actually kind of the flip side. So this is the typical, not always, but in most cases of hypoplastic left heart and Jacobsen syndrome, you see just this little slit. And instead of this massively thickened ventricular wall, we see this kind of what we call a slit-like left ventricle. Okay, and that's actually what we're focusing on. But in this case, I think in contrast to that, this is probably more due to decreased lack of cardiac myocyte proliferation. And that's what I'm going to tell you a lot about in the rest of my talk. Anybody have any questions? I know I'm throwing out all this stuff, but basically what this leads us again to believe is that somehow as the heart is developing, there's a lack of growth. And
I
that's,
have a question. before
Yeah.
you get that. Are each of those 4 pictures the same, like, view,
yeah, so same magnification. These are pathologic specimens.
but it's coming from the same angle to that part of the heart?
Well, actually, they're oriented differently, right? So, yeah. Although I guess you could say that the 2 on the left, well, yeah, they're angles related. But it's still pretty much a comparable comparison, just to highlight the most obvious features of that distinguish these. Yeah. Okay, so like I said, in some cases, like what I showed you, it may be due to little growth, and in other cases, too much growth. But if you're going to try to understand the mechanism, you better appreciate those differences to explain it, right? Okay, so this takes me back to literally July of 1995, 31 years ago now. This was Edgar when I first met him, when I was in my first month of training, and this was what was in his paper chart showing the chromosome 11 deletion, which I think all of you can appreciate. And all I knew is there must be something interesting, right, in this region that's lost on the affected chromosome, but I had no idea. You have to realize back then we didn't have any of these amazing technologies that we have now. We didn't even know, maybe a little bit, some of the genes in there, but I certainly didn't know by looking at that, which genes were actually affected. So that was going to be a big first challenge, something that probably took us close to 10 years is to actually identify a gene. These days, the technologies we have now, we could probably identify what we call candidate genes in a matter of days, which is 10 years, just to give you an idea of the progress. So you've already heard a lot about these 3 problems. So fortunately, because of people like Chuck Haymeyer, we've come up with the protocols, and thanks to our medical experts like Virgil and Rennie Favier, that I'll talk about in my next talk. Bleeding and infection which has historically have been among the most common causes of death, really should be now history, and hopefully never to see that happen again. The challenge is to get that information out, obviously. So that leaves us still with heart defects, potentially as the most common cause of death in infants. So to attest to our 2 stars here, if you make it past the first year with hypoplastic left heart syndrome, where you've probably already had 2 to 3 palliative surgeries, you're most likely going to do pretty well for quite a long time. Not a cakewalk, but if you survive the first year, you're going to be well on your way into adulthood, as exemplified there. Again, it's so cool to actually personalize my talk to history in the making. Okay, so I spent the first part of my research just trying to learn everything we could. And as I mentioned on Monday, we published this paper in 2004, characterizing 110 cases. The next biggest study prior to ours was 17. So we learned a lot, obviously. And this is actually a real human heart, a baby that passed away at age 13 days. With Jacobsen syndrome and hypoplastic left heart, and I don't think I have to convince you how abnormal that was supposed to be the left ventricular chamber. And I don't think it's a hard sell to convince you that that was not going to be compatible with life. And I'm going to come back to this heart because this is one of the hearts that the family donated to us, and we've actually been able to make some really exciting discoveries thanks to this. Okay, so again, one of the challenges is to try to understand whatever the gene or genes are, how do they cause these different kinds of heart defects? My focus for today is going to be mostly on hypoplastic left heart, but I will touch on some of the others along the way. So this is one of the big technologies that really catapulted our research, because that paratype that I showed you before is kind of like, I compare it to like looking at the stars at night with a naked eye. You can see them, but you really can't resolve anything. This allows you now to like be looking through the Hubble telescope to really resolve the deletion breakpoints to the individual gene level. And that's really important because a lot of you are obviously wondering, like, what does the future hold for your child? There are exceptions, but generally there is a pretty good correlation between deletion size, which genes are deleted, as you can imagine, and what to expect in terms of outcomes in the future. So this is what people call microarray, what was referred to yesterday. It's actually smaller than that now, but this little tiny grid that has a ton of probes that allow us to map exactly, like you saw in the talk yesterday, yesterday or Monday, yesterday, I guess. You know, whether there's the normal number too much or too. So this is what that's all about. And this is all done robotically with computers, so it's pretty simple in terms of labor intensity compared to when they showed me the karyotype analysis. That's much more labor intensive, and to get trained to actually analyze those chromosomes typically is about a 2-year training program. So that's kind of falling by the wayside, unfortunately. What we could do is actually not just for clinical purposes, but for research purposes, determine what genes are in the region that's deleted in, let's say, a child with Jacobsen syndrome or hypoplastic left heart. And what we were able to take advantage of is, even though, as shown here in blue, this is kind of a typical schematic diagram of a deletion that goes out to the end of the chromosome. So that's why it's called the 11q terminal deletion disorder, to determine the end of the chromosome. But we also already knew of a number, less commonly, but some cases where maybe a child with hypoplastic left heart syndrome who has a deletion within chromosome 11, but not extending out to the end. And so we call that an interstitial deletion. And even rarer than the terminal deletions, we could take advantage of these patients with these interstitial deletions with hypoplastic left heart syndrome that would allow us, if it's the same genetic cause, to hypothesize that it might be in this region of overlap, right, between the blue and the red for hypoplastic left heart. And then in the early 2000s, when the Human Genome Sequencing Project came out, we could know exactly which genes are in that now what we call critical or minimal region in chromosome 11 for the heart gene, or heart genes. And we actually identified 2 genes. The first one is the one I'll talk to you mostly about today, just because of history, ETS1, which already had been shown in some, at least one other animal model system, to have a critical role in the development of the heart, at least in an organism called the C-squirt. And more recently, we think that Fly1, which is another family member of ETS1, also probably has a critical role. And these 2 genes function as what we call transcription factors. So they are molecular switches that turn on and turn off other genes to do their function, either during critical times of heart development for normal function, and in some forms of cancers, I mentioned to you before, they are abnormally expressed and turn on and off genes that can lead to cancer. So, and we're pretty sure that these other 4 genes probably are not relevant in terms of causal growth. Let's see, there we go. And just to make it a little more compelling, we actually came across a patient a number of years ago, not with Jacobsen syndrome, but with a complex heart defect, including an abnormal left ventricle, who rather than having a deletion, just had a single point mutation in the ETS1 gene. So that added more evidence to suggest that, in fact, ETS1 is at least one cause. And kind of parenthetically, we have a similar scenario now for Fly. So that's why our most recent thinking is the 2 genes work together. Hopefully, there we go. Talking about how the 2 probably work together. And again, just to reiterate, in most cases, like in the case of this patient with the Fly1 mutation, that patient has the same exact form of hypoplastic left heart syndrome that occurs most commonly in Jacobsen syndrome. So like I mentioned to you before, we use mice, and all you need to know from this picture is wherever the blue is, that's where ETS1 is expressing or working in the developing mouse heart. And what that allowed us to determine is that there are these 2 populations of cells that are essential as the heart develops, called the cardiac endothelium, which I'll tell you about, and the neurocrust. And basically that led us to hypothesize that loss of ETS1 in one or both of these populations of cells may give rise to one or more of the different heart defects. For any individual, it's a little bit hard to predict which it might be, and we might have time to come back to that. And again, if we're talking about hypoplastic left heart syndrome, if it's a problem with how those cardiac myocytes are growing and developing to form a ventricle, it's actually not directly due to an effect on cardiac myocytes, but it's through an indirect effect from the other cells that are expressing ETS1 that somehow affect how cardiac myocytes grow over time to form the ventricle. All right, so this is a really commonly used cartoon picture showing you what the cardiac neurocrest is. And what it is that as the fetus is developing, there is the neural tube, which becomes the spinal cord. And there's actually a population of cells in this developing neural tube that actually leave that developing neural tube and have to migrate to the developing heart to ultimately form some of the permanent structures of the heart and some of the vessels. So the vessels, the valves can actually be outflow tracts of the aorta and pulmonary artery and that wall that separates them. So it's actually an amazing system that somehow has evolved this population of cells and subpopulation of cells that originate from basically the developing spinal cord to form, again, some of the spinal structures in the heart. And as a little hint, we know that there was a study, this study was published a number of years ago by a surgical group that showed that in patients with all comers with hypoplastic left heart syndrome, not Jacobsen syndrome, there is a much higher frequency of something called Hirschsprung's disease, which is an intestinal motility defect that is due to a neurocrest defect. And so that is an association that suggested that neurocrest dysfunction may have some component in the pathogenesis of at least some patients with hypoplastic left heart syndrome. The problem though is that to this day, whenever we do a knockout of the ETS1 gene in mouse, we have never seen hypoplastic left heart syndrome. Now I'll come back to that, but what we do see is something that some of your kids have been born with called ventricular septal defect, and specifically something called double outlet right ventricle. So if you look at a normal, this is actually a three-dimensional cardiac MRI of a mouse heart. And what you see here is the left ventricle, the right ventricle. I don't know if you can see that little black, that's the wall that separates the two chambers. And so that's called the interventricular septum. And the red here is the aorta, as you can see, aligned with the left ventricle. And in our ETS1 knockout mice, what we see is rather than this ventricular septum intact, there's a hole. So that's why we call it a ventricular septal defect. And we call it double outlet right ventricle in this case, because now what we see going along with that hole is a shift in how the aorta is hooked up to the heart. It's now hooked up to the right side. So both the aorta and the pulmonary artery come off of the right ventricle, along with the hole in the heart. So that's how we come up with the term double outlet right ventricle. And fortunately, in most cases, it's a one and done. You close the VSD and kind of redirect the aorta to the left ventricle, and hopefully that's the only surgery you need. In other cases, though, in our mice and certainly in humans, going along with the septal defect and the alignment problem, the aortic valve, which normally has three nice leaflets here, is pretty significantly altered so that instead of the normal three leaflets, you now see only two leaflets. And aortic valve problems can be more challenging, requiring in worst case scenarios replacements and whatnot. So that's a bit more challenging. Now, in terms of what happens, remember I showed you those cells have to migrate to get to where they need to go. And in mice, what we could show really beautifully, thanks to one of the PhDs in my lab at the time, is that normally, just like in that cartoon, what you see here, this is a little part of the neural tube, and what you see blown up is those neural crest cells that are labeled here specifically migrating to get to those structures that are forming in the heart. And in our mice in which we've knocked out ETS1, instead that whole stream of nicely migrating cells is disrupted. And so we think it's at least in part due to a migration problem that prevents the formation of those structures. And specifically what we're talking about, here's the cells that actually make up that septum. And what we've been able to show, it's clearly a defect in the ability of those cells to migrate to get to that septum. All right, so that's kind of where I'm going to leave you in terms of the neurocrest story. It's great, we have a beautiful paper that came out, but it didn't get us to where I want to go in terms of understanding faith and part of this defect, right? So this is just to remind you of what I alluded to you before, that in the actual heart, there are two populations of these so-called cardiac endothelial cells. So one is what's called the endocardium, and that, if you can see here, this little green is the inner lining of that wall. And then there's the other population of endothelial cells in the heart that make up this kind of outer layer called the compact zone. And in that compact zone normally are little tiny branches of the coronary artery. So we call that the coronary vascular endothelium. So cells that make up those little blood vessels that are critical to promote the growth of this compact zone. And it's actually this compact zone that really does most of the work when the heart contracts. What you also see here, if you look carefully, is these little finger-like projections. That is another component of the ventricular wall as the heart is developing, and that's called the trabecular myocardium. The mature heart, the trabecular myocardium goes away. And what it actually is doing is through all these little finger-like projections, before the coronary arteries are developed, these finger-like projections increase the surface area to allow the components of the blood to be absorbed to feed and supply the nutrients for that developing compact zone myocardium. So that's called the trabecular layer, which kind of goes along to the right early on, but does have a very important function, which I'll come back to in a minute. In addition, probably to help the ventricle form. And once the ventricle is formed, you just kind of wind up with basically a nice smooth layer of compact zone. Okay, and so just to show you this in our mice, compared to a normal mouse heart, and so this is that compact zone I was telling you about, and these are kind of like those finger-like projections. If you delete ETS1, in the most severe case, you see now a complete loss of that compact zone, and you just see this very lacy-like kind of trabecular myocardium. And this is actually from a mouse embryo that didn't even survive to birth, because you can imagine if you lose this compact zone, which is what allows the heart to squeeze, you're not going to live very long. So that was a really important observation, now implicating for the first time X1 in regulating cardiac endothelium. And I'm going to continue on that theme in a moment. And so what we also were able to show here is not, well, let's see here. If you look at an enlargement of a normal heart and then the mutant heart, this is the compact zone, and you're probably not going to see this very well. But what you see is a nice layer, and there's little red outlines. Those are the coronary vascular endothelial cells. And in the mutant heart, what we see, just like what I kind of showed you before, is that compact zone now is thinner, and there's almost a complete loss of those little red signals of those coronary vascular endothelium. This is just kind of another way of quantifying that. And if you look grossly compared to a normal heart, the coronary arteries are less formed, less complex, less branching. And so we think at least part of what's going on in the endothelium is that S1 is required for these endothelial cells to help the coronary vasculature develop, which is then required to help that compact cell develop. And I'm going to come back to that in a moment, but what we were able to find is that through some very sophisticated technology, we could actually then identify some of the different genes in those endothelial cells that ETS1 is regulating. And it turns out this number of genes that are directly targeted by ETS1 are all involved in the development of the coronary endothelium. So that all makes really good sense. And interestingly, if you look at, for example, this first gene, ALK1, which has another name, it turns out mutations in that gene have been associated with total anomalous pulmonary veins, which is one of the heart defects that occurs in Jacobsen syndrome. Versus other patients, well, other genes cause other heart defects. So for any one patient with Jacobsen syndrome who has some kind of a heart defect, it's kind of like a roll of, playing craps. It's a kind of a roll of the genetic dice to see if you're more sensitive to its effect on this gene, which leads to pulmonary venous anomalies, let's say, versus this gene, which leads to something called pathology. So we definitely made some advances in understanding how different heart defects occur in different patients, even potentially with the same relation. So intuitively, you can imagine, remember I showed you that compact zone is thinner, and so it's not going to work as well. What this shows you, don't worry about the details, is that it's actually in the absence of ETS1, it's actually due to decreased heart cell growth. So cardiac myocyte proliferation is decreased. And that led to a few more really interesting observations. Part of the mechanism is shown here is that in a normal mouse heart, if you see this white dotted line, this is just showing you how as the heart is developing, those coronary vascular endothelial cells, they kind of start up here at the top of the heart. And they, over time, migrate down to get ultimately down to the bottom of the heart. When we eliminate PTS1, those heart cells fail in their ability to get all the way down to the bottom to fill up that compact zone that's developing and needs signaling from those cells to grow. And this is another way just to show that any of these genes that we showed that are regulated by ETS1 that are required for these blood vessels to form, we can actually do sort of in vitro studies to show that any one of these genes also affects the development of those coronary arteries. So ETS1 and or any of these genes that it directly turns on or off are critical for these blood vessels to form that are required for the heart to develop, in a nutshell. Now, taking it one step further, we most recently have been able to perform an analysis called single cell RNA sequencing, where we can actually take an individual cell from either a normal heart or the mutant heart and analyze the expression of all 23,000 human genes in one cell. And we can do thousands of these cells. And what Lou Wayne in my lab, who is an absolute superstar, so whenever I ask you for money, it's basically so I can keep him employed. What Lou Wayne has shown is that single cell RNA sequencing showed that when you knock it out in the coronary vascular endothelium, there is a loss, of 3 factors that are secreted by the coronary vascular cells. And these factors promote heart cell growth, cardiac myocyte proliferation. Does that make sense? So remember when I showed you before, hypoplastic left heart syndrome and Jacobsen syndrome is probably due to decreased growth of that developing ventricle. And now we think we have a mechanism that shows how that happens. Loss of ETS1 affects the development of the coronary vasculature, which prohibits the normal secretion of factors that are required for growth. And this is just to show that if you can add back those factors, not adding back ETS1, that would be gene therapy. This is what I would call gene replacement therapy. You can actually now rescue that thin compound zone and allow it to grow back to being normal. Not only in the mutant heart, but also in this case, this is just to show you that you can take a normal heart and treat those heart cells with these same factors and promote growth and proliferation of normal heart cells. Why is that interesting? What happens when you have a heart attack? If I go to the ER right now with chest pain, I'm killing off heart cells, right? And normally when you have a heart attack, those heart cells are replaced by scar tissue. So the big challenge in the world of adult cardiology is to figure out if there's a way that we can prevent scarring and instead promote replacement of killed off heart cells with new hearts, what people refer to as cardiac regeneration. It's early on in the game, but we're very excited that not just for hyperplastic left heart syndrome, but these same growth factors may have the potential to treat adults having a myocardial infarction. So remember what I told you, even though we're starting with something rare, maybe it has broader application. Well, this goes beyond my wildest dreams.
Does that mean that you could also put those genes into Faith and Harlan now?
Well, that is a great question. If everyone heard, we promote their left ventricles to grow. Well, that's what we have to figure out, right? I just showed you in a mouse embryonic heart. I don't know if it's going to work in a 25-year-old human heart, but those are exactly the studies that we are aiming to do next
But you could
with
potentially do it-
financial support.
Yeah, but you could potentially do it in babies. Like,
Yeah,
it's too late for
eventually.
them. They're too
Now,
late.
yeah, and there are kind of intermediate cases where, you know, if the valves are completely blocked off, that's more difficult. You can't create a new valve very easily. But one of the things that people think is if you can promote growth when there's still some flow, you know, a kind of less severe form, then that might be a population that could be targeted for this kind of therapy. Stay tuned. That's pretty good. All right. And this is just to kind of summarize what I told you, and I didn't tell you that the endocardium we also showed, and others have previously shown, secretes a whole pathway involving signaling that promotes cardiac myocyte proliferation. But what Lou has shown is that these two of those factors that he identified had never been discovered in terms of their ability to promote cardiac myocyte proliferation. So yes, we're kind of giddy with excitement, and this is why I say after 30 years, you know, I'm just getting started. This is so exciting. Okay, so that's what we know about in the mouse. And as I mentioned to you before, there's some limitations and some strengths, but sometimes you want to use relatively simpler organisms to study other aspects. And in this case, this is just an example of what happens in a frog heart, which is a little bit different, but some similarities to a human or mouse heart. And what I'm showing you here is just a cross section of the ventricle of the frog heart, and you can kind of see this outer layer and then these kind of finger-like projections. So that's the frog version of that complex zone and the trabecular myocardium. And we can do the same kind of genetic tricks to knock out ETS1 in the frog heart. And what you see here, compared to that normal ventricle, is in this case, not that what we call non-compaction that I showed you, but here, this is a truly small left ventricle. So it's a hypoplastic ventricle. And you see these nice little finger-like projections there, the trabecular myocardium, completely gone. So in this case, we actually do get kind of the frog version of hypoplastic left heart syndrome. And that is very exciting because at the end of the day, why do we think in frog we can get something like a hypoplastic ventricle versus in mouse, we only get that other type of phenotype? And the answer we think is that in the higher systems like mouse or humans, you need both ETS1 and FLY1. And that's why we just spent $23,000 to genetically engineer a mouse that has just those two genes. So stand by next conference, I hope long before then, to give you an update on that. The other thing that's really cool, kind of alluding to your question before, is in frog, my colleague at Georgia Tech, Shuey Nee, is an embryologist, and one of the things that we'd like to do is transplant experiments, and what she could do is actually transplant the progenitor cells for those endothelial cells and whatnot to the mutant frog heart. And what she showed in a frog heart is that you can actually prevent or rescue that hypoplastic ventricle if you transplant those progenitor heart cells early on. Now, obviously, that doesn't translate directly now to a human heart, but it does suggest the possibility that as the human heart is developing, if you could identify hypoplastic left heart syndrome very early on, there could be a window of opportunity to intervene early on to prevent that from happening. That's a little bit science fiction now, but that's something obviously in the future that would be really exciting. So again, we started with a rare syndrome, and now we're talking about even a broader application. All right, so my last little bit of time, I wanted to show you the studies that we've been doing for the last 3 years now from this human hypoplastic left heart from a 13-day-old baby that was born with Jacobsen Syndrome. And again, this is that very, very abnormal slit-like left ventricle. And what we've been able to do, I know this is going to be hard to see. Can we turn down the lights a little bit more? Because this is actually really cool. What you're going to see on the left is from a 13-day-old baby that died from non-cardiac causes, an analysis of the heart cells. Got to make it really dark. Put everybody to sleep. Come on. You're fired. Well, what I'm going to show you, you probably can't see it unless you come up here. I can't even see it from here. Is there the apparatus in the heart cells that actually causes the cell to actually contract, and all the cells contract. That's much better. One more. One more. Those are off. Don't give me that. Anyway, if you could come up here closely, what you would see is it looks like railroad tracks. And those railroad tracks are actually the sort of functioning unit in the heart cell called the sarcomere that allows the heart cell to contract. And both in the left and right ventricle of a normal heart, you see this very nice organization. And basically what you see in that hypoplastic left heart is a couple of things, including a much higher density of heart cells. If you look at these blue dots compared to the control, there are a lot more blue dots. That's just labeling the nucleus of the heart cells. And it looks like someone detonated a hand grenade in there, because now instead of this nice railroad track organization at the sarcomeres, is kind of obliterated all over the place. So it's these kinds of studies that are for the first time really giving us some molecular and cellular novel insights into how you get hypoplastic left heart syndrome. And one more thing that we've observed, which comes back to what we actually have seen in the mouse heart, is you're gonna have to kind of take this for on faith here, but compared to a normal heart, all these red dots here are actually those endothelial cells that make up the coronary vasculature in that heart muscle. And what we see here is a little bit hard to tell, is there is actually these little red dots here, these red outlines are actually in the blood vessels. These are actually just little red blood cells themselves. And what we actually see is a dramatic decrease in those coronary vascular endothelial cells in that hypoplastic left heart. So that is consistent with our working model that X1 and probably Fly1 together are regulating how that coronary vascular endothelium helps to form those blood vessels in the developing heart, and loss of X1 results in the loss of those growth factors and components. So I'm going to come back to that in a little bit more formal way in a second, but this is just kind of reiterating what I just told you. So this is just coming back to that early slide that I showed you. I think I kind of rehashed it, but I kind of went ahead on myself. Moving forward, so I wanted to just kind of articulate it in a little more detail what our current hypothesis is. So what I showed you in the mouse, we get overgrowth of the trabecular myocardium and a thinning in the compact zone. In the frog, we see a little bit different situation where we see a loss of the trabecular myocardium and then this kind of high density ball of cells that are part of a very small abnormal ventricle. What we don't know is why the difference between species, right? So that's really interesting. But I think the fact that in one type of heart defect, you see overgrowth of that trabecular myocardium, and in the hypoplastic ventricle, you see an absence of that trabecular myocardium, strongly implicates the trabecular myocardium as having some causal role in whatever effect you have on the developing ventricle. And that's actually something now that we're very actively pursuing. So in 30 years, this is what we're left with at this point. So I didn't really talk about this today, but part of the defect is also an effect on the valve, the mitral valve, that has to open normally to allow blood to come into that developing ventricle. Well, part of the developing mitral valve includes endothelial cells, like an endocardium, believe it. And so in Jacobsen syndrome, we know most likely very early on, and in other non-Jacobsen syndrome patients, that the mitral valve is very abnormal, so you get impaired blood flow into that developing ventricle. And we know already from other experiments that you need normal flow to help promote growth and development. What I've shown you today is that when you compromise the endothelial cells, like when you lose DPS1, you affect actually two populations, the endothelial cells that I just showed you that secrete those factors, as well as that other population of cells that I didn't really spend much time talking about, the endocardium, which also secrete factors. And lastly, which I didn't really have much time to talk about, is this association of the trabecular myocardium. And it turns out that for the trabecular myocardium to form, there are factors that are in the region or the space between cells called the extracellular matrix. And it's people are just learning about this, but the extracellular matrix is just kind of like this jello mix of factors that actually are, as we learn more about it, are very precisely regulated to make sure that as the ventricle is developing, the heart cells line up in the correct orientation and allow for that ventricle to grow out and form normally. So we think that when you lose the trabecular myocardium, that is also required for that ventricle to align in the right way to normal. And one of the studies we're actually going to be starting very soon is to specifically take out the function of the trabecular myocardium at certain stages of heart development to see what effect that alone has. We predict that the trabecular myocardium has a critical role that, when abnormal, may lead to a hypoplastic ventricle. Okay, so just to repeat myself again, stating the obvious, we wouldn't be here today without so many people here and elsewhere that have done amazing things to support my research. I talked about Fred and Rebecca, who again, alone in the last like 18 years have raised something like $2 million for my research. So I jokingly say without them, I would have a one slide presentation. It would be a white slide. So so much of this is because of theirs and the support of other groups, the 11Q group, 11Q Spain, Europe, and some of these other foundations that more recently have been saviors. And I also really want to acknowledge, there's a whole team that works together. Lou Wayne is this amazing, brilliant PhD scientist who came to my lab originally about 8 years ago from China. His English is better than mine, I swear. And he does the work of 3 people, and he has really taken my research to a whole nother level now. And so without him, there would be nothing. In addition, I am really, really fortunate to have, again, as part of this sort of multidisciplinary team, I'm the MD with a science background, and I work with these 3 PhDs, Karen and Rolf, a husband and wife, who work on heart development in the fruit fly and the zebrafish. Some of you have heard me talk in the past about how many of the same genes involved in the heart development of a human are in a fruit fly. And literally, some of their work on fruit fly heart development has allowed me to implement life-saving strategies in some of my human patients with congenital heart disease. So I always make it a point, and next time you see a fruit fly flying around in the kitchen, don't kill it. Get out of these things. And then Shui Ni is another incredible PhD scientist whose work a little bit I showed you on the frog part, who actually have had the wherewithal to realize that there actually could be a window of opportunity to prevent hypoplastic left heart. And so this group of people I'm so blessed to have as part of our team. And so when I ask you to support my research, this is what you're supporting. And sadly, I've mentioned some of these kids before. Every one of these babies or kids has had a major contribution. It's not linear. You have these quantum leaps, you have these rare individual patients that might give you some insights. And all 7 of these babies have, and sadly, the price is all 7 of these babies of these patients are now deceased. But this is kind of part of their legacy, and that's why I want to share that with you. And I know that, their families in all cases have been true heroes to step up in their times of, total tragedy to want to help families like you. So again, when I'm asking for support, a lot of that comes from what these families have done as well. And finally, I do want to revisit Edgar. Again, I keep sounding like a broken record. It's literally 31 years ago, practically like today, that I first met Edgar. And, we've had some early discussions. I would love to be able to maybe start some kind of a scholarship fund for the high school where he went to in San Diego to support students that maybe are wanting to do something to help people with special needs. I know there's some legal, obstacles to that, but I have discussed this with Edgar's family, and they would love to see some kind of like a small scholarship fund to help promote future people that help children like yours. And I think that's about it. Oops. Yes, I'm done. So any questions? Okay, hang on. Yeah, go ahead.
I just wanted to know, so for children with Jacobsen syndrome who are born without a heart defect,
Yeah.
that's like their first cardiology
Yeah.
evaluation, like Shelby, for example, she's a heart murmur. Should she be going back? Should she be following up like within 5 years to check on her heart?
Right, so obviously she's going to see her pediatrician every year. And assuming the pediatrician thinks that she's doing fine, in the absence of any new symptoms or new physical findings, like a new heart murmur, my recommendation is every 5 years,
Oh, okay.
just kind of for surveillance.
She never even thought about heart murmur, it was the cardiologist.
Well, of course, right? But I think every 5 years, if they're otherwise doing well with a previously normal echocardiogram, because that defect that I told you about, called non-compaction with the thing in that wall, I showed you the most severe form. It actually can develop in adults. Rare, but there are a couple of cases in Jacobsen syndrome, and I don't want to miss that. And so it's better to, catch something like that early. So if they're doing fine, with a previously normal echo, I take once every.
Thank you. Thank you very much.
While,
I'm just wondering, I don't need that, thanks. I'm just.
well, actually, for anyone who's online,
It helps them here. Oh, I.
they want to hear you.
Okay, well, I won't speak. No, I'm just wondering if any of this research I know about growing, potentially growing the left ventricle wouldn't work for somebody who's already been plumbed. Is there any of this that could create something to help strengthen the?
Yeah, so that's a great question. The other thing that we might be able to do is, I'm sure this is not news to you. When Faith and Parliament had gone through the 3 surgical stages, basically to reconnect the plumbing, what they're actually doing is, at this point, the left ventricle is unusable. I got convinced of that. So what they actually do is connect the right ventricle to the aorta so that the right ventricle actually becomes the main pumping chamber of the heart. The problem is Mother Nature did not normally design the right ventricle to do the work of the left ventricle for the next 80 or 90 years. To answer your question, one possibility may be that over time, we know that not everyone, but in a lot of cases, as people with hypoplastic left heart syndrome get older into their adult years, that right ventricle may get tighter. And so to your question, one possible strategy might be to treat that right ventricle tissue with these factors to promote more heart cell regeneration and proliferation. So that basically increase the muscle to help it.
Like,
That's
somebody with a heart attack. then you
right.
get-
Now, one of the challenges though is that in general, we know that the ability of the human heart to regenerate is very, very limited after maybe the first year of life. And we don't know why. And that's kind of the holy grail of adult cardiology and even congenital heart disease. I actually think there's also for like heart attack, there's an inflammatory component. So you might need, in that case, something that promotes heart cell proliferation and something that shuts down the inflammatory response, because that's what leads to the scarring. So there are actually some studies out there that hint that like steroids may help some patients suffering a heart attack. And I think it may be like this 1-2 punch, promote growth, prevent scarring. So it's all out there. I just need money to keep it going, but yeah, I know I'm broken, but it's exciting, right? I mean, we couldn't have even had this discussion 5 years ago. So that's pretty cool, right? Any other questions? Yeah, Jeremy.
Yeah, you kind of already touched on it a little bit, but has there been any, are there progressive, things? You kind of touched on it with future, options and all that, but are there progressive things with the ETS or the Fly One that either hypoplas or any other defect, is there any research on any progressive problems? Not just at birth.
Well, so the cardiomyopathy, right? So what I showed you is actually the third most common form of cardiomyopathy in humans. It's called non-compaction, for inaccurate reasons. So, again, our studies are in a mouse embryonic heart. But let's say one of your kids is 15 years old and starts showing signs of this thinning, right, of the compact zone with decreased function and overgrowth of extraventricular myocardium. So in theory, the other thing that we're looking at, and I've actually started having some very early discussions with something called the Dilated Cardiomyopathy Foundation to see if they might be interested in helping us to, I think, do exactly what you proposed, to see if we might be able to target a subset of patients with dilated cardiomyopathy to get that heart tissue to grow to overcome that thinning and loss of function. Does that kind of answer your question? Yes. Stay tuned.